Hi everyone, thanks for checking out this video. The last video was like an optional review of high school biology, covering some topics that I didn't want to completely leave out, but aren't really like that necessary for this course. But in this video, we're going to be talking about the like foundational concepts in molecular biology that will actually be like really important and really give us like a foundation to work with to start doing this like bioinformatics analyses we're going to do. So if you guys have been watching this series so far, hopefully you remember this from the first video. I kind of touched on this at the very end of the first video, introducing the course. So yeah, just to repeat from that, this diagram is showing how information can be stored in stretches of the DNA called genes, and that information can be used to produce strands of messenger RNA that are then used to put together proteins out of amino acids, and then the proteins actually do a lot of the actual work of the cell. They're involved in like signaling, cell cycle control, stimulus response, metabolism, regulating various like cellular processes. So yeah, the proteins do like the actual work and like functions of the cell, and they're produced from information that's stored in stretches of DNA called the genes, and they're produced through this intermediate step of creating messenger RNA. And this process where information flows from genes in the DNA to the mRNA to the proteins is called the central dogma of molecular biology. And you guys might hear people saying sometimes that the central dogma has been like debunked or disproven. When people say that, they're playing a little bit of like a language game. Like it's a bit of a semantic argument people sometimes get in, just debating like semantics. But yeah, when they say it's been debunked or disproven, what they actually mean is that there's been exceptions found to it. So up to you guys, however you want to define the word debunked, if finding exceptions to something counts as debunking it. Personally, I would not say that it's been debunked or disproven. I would say that it's an extremely useful model that's very useful for understanding biology at the cellular and molecular level. But yeah, there have been exceptions found to it. So it's not a perfect model that's true 100% of the time, and it maybe has less explanatory power than when it was originally proposed. But yeah, that's a bit of just a semantic debate. When people say it's been like debunked, yeah, I would not say that. But yeah, what people mean is there's been exceptions found. And so rather than telling you guys some of the exceptions, I'm actually going to put the question to you guys. Do any of you know of any exceptions to this flow of information? Is there any situation you guys can think of where information does not flow this way in cells at the molecular level? If you guys know of any exceptions, please leave a comment and tell me what exceptions you guys know of. I want to kind of put the question to you guys on this, if you know of any exceptions to this flow of information in cells. And yeah, so just to give you guys another like piece of terminology here, this process is also called gene expression. So if you guys hear this term gene expression, it means the process by which information encoded in the DNA is used to make a product. Meaning mRNA or a protein. So if a gene is being expressed, that means the information in the DNA is being used to actually create something that is then like physically like floating around in the cell or like performing some function or just yeah, it's the information is being used to actually create a product basically. And this can be interesting because different gene expression patterns could be signs of like interesting biological differences between cells. So you could think about like a given gene, and you could talk about and then you could ask how the expression of that gene differs between different kinds of cells. Like, for example, maybe a gene has higher or lower expression in like cancerous lung cells compared to healthy lung cells. So yeah, like differences in gene expression can be signs of interesting biological differences that we might want to study and learn more about. And yeah, again, just reviewing some terminology from the first video. So the process where mRNA is created from the information in the DNA is called transcription. The process where the protein is created from the mRNA is called translation. And you might hear the strands of messenger RNA also referred to as transcripts. So that's where the word transcriptomics comes from. If you guys remember, this omics suffix just means the big data study of something. And we can talk about like genomics being the big data study of genes, proteomics being the big data study of proteins, and transcriptomics being the big data study of the transcripts, or these strands of messenger RNA. And when we're putting together this like mental model of things that are happening in cells at the molecular level, it can be useful to think of the DNA as just being like stable and being there and just existing in the cell. And on the other hand, we can think of the mRNA and the proteins as being like semi-stable, but also like semi-transient. So they're produced from the gene, and they kind of float around in the cell, and they float around a bit, do their thing a bit, but they also break down and disappear over time. So the process of gene expression can be kind of like thought of and modeled as a random stochastic process, where the strands of messenger RNA are being produced from the gene and kind of floating around and existing in the cell for a bit, but also breaking down and disappearing over time. So this circle with a line through it is like how we show the breaking down of something and the disappearance of something in these like diagrams. So yeah, the mRNA is produced from the gene. These strands kind of float around in the cell and exist in the cell for a bit, and they also break down over time. And we can model the production and the degradation or the breakdown of them as both being like random stochastic processes. And same with the proteins. The proteins usually, not 100% of the time, but usually are more stable than the mRNA. So they have like a longer lifetime than the mRNA strands. But they're still semi-transient and like semi-stable, semi-transient. So they still are produced in like a random process using the strands of messenger RNA. They're produced and they kind of float around in the cell a bit and also break down and disappear over time. So again, the production and the degradation or the breakdown of the protein can also be modeled as like a random stochastic process. And something else important is that organisms have many different genes. So before I was showing you guys like a diagram of one gene, but organisms have many different genes that produce different proteins that do different things. So humans, for example, have about 20,000 or so genes. And yeah, different genes produce different types of strands of messenger RNA, which produce different proteins, which have different types of functions, basically. And sometimes when we're trying to make like a mental model of something, it can be useful to have simplifications just to help us like think and reason about things. And that can be useful even if they're not like literally 100% true. So a simplification I'm going to give you guys that's not 100% true, but that will be useful as like a mental tool to think about these things, is that it can be helpful to think about this as if there's a 1-to-1-to-1 relationship between a gene, the type of mRNA produced by that gene, and the type of protein that's ultimately produced. So it can be useful to have like a mental model of the there being like a 1-to-1-to-1 relationship where every gene produces a unique type of messenger RNA strand, and every messenger RNA strand produces a unique type of protein. That's actually not literally true. It's actually the case that different types of proteins can be produced from the same gene based on all kinds of different like more complicated processes, including different ways the mRNA can be cut up and spliced together in different ways before translation, different like start and stop sites of transcription in the DNA, and even things like extra processing that happens to proteins after they've already been translated from the messenger RNA. So there's actually all kinds of different ways that different types of proteins can be produced from the same gene. And those proteins are usually called isoforms. And a lot of the time they have similar functions. But yeah, that's the reality. The reality is that actually different types of proteins can be produced from the same gene based on these like complicated ways that things can be processed at different steps. But for the purposes of this course and for the purposes of simplifying to have a useful mental model, I think it can be a useful simplification to think as if there's like a 1-to-1-to-1 correspondence between these things. And actually, the way biologists talk about things, they can sometimes get a bit loose with their phrasing and talk about things in a way that actually does kind of like make this simplifying assumption. So for example, if you hear people talking about the p53 tumor suppressor protein, which is a common type of protein involved in a lot of types of cancer. If you hear people say like p53, for example, they could be talking about the actual protein itself, or they could be talking about the gene in the DNA that contains the instructions to make that protein, or they might be talking about the corresponding type of messenger RNA that's produced from that gene. And yeah, if they were being like actually technically precise with their wording, they would be calling the actual stretch of DNA that makes up the gene TP53, and same with the corresponding mRNA. But yeah, just in real life, the way people talk about things, yeah, they'll use the word P53 to refer to the actual P53 protein, as well as the gene that contains the information to make that protein, as well as the type of messenger RNA that's the intermediate step that contains that information. So yeah, and just based on the context, you kind of can figure out if they're talking about the protein or the gene or the mRNA. And yeah, we talked before about like different functions a protein can have. It can be involved in like metabolism or like signaling or stimulus response. Well, one of the possible functions that a protein can have is actually to regulate the expression of other genes or even of their own gene. So you could imagine a protein, and yeah, these are called transcription factors. You can imagine these transcription factor proteins binding to the DNA close to the site of another gene, and then making it easier or harder for mRNA to be produced from that second gene. So yeah, we call this like gene regulation, where the protein from one gene can regulate the expression of another gene or of itself. And actually, this diagram is only showing like a situation where one protein is regulating the expression of another gene, but actually these can form like pretty complex networks where you can have entire networks of genes all like regulating each other basically. And yeah, they can get to be pretty complex networks. And actually the field of gene regulatory networks is like itself like a whole field of study of like how to measure them, how to model them, how to infer when the protein from one gene is involved in regulating another gene, how to like figure out when that's happening. And yeah, they can form like pretty complex networks that can actually be pretty important for all kinds of biological functions. And when these networks go wrong or like something goes awry in them, that can lead to diseases like cancer if things are not being regulated properly. But yeah, so just to like make one last point on this slide, when you think about this like central dogma process of genes being used to produce mRNA, which is then used to produce proteins. And then you combine that with the fact that these proteins can regulate the expression of other genes or of themselves. That model is already enough to get you pretty far in explaining a lot of like cellular complexity. So just having the model of like gene expression and gene regulation, that model, when you start to think about that in complex networks involving like dozens or hundreds or thousands of genes, that can get you pretty far in starting to like model and explain a lot of complex things that that cells do, basically. So yeah, we've started with this central dogma, and yeah, we now have something like a framework that we can use to understand pretty complex things going on in cells, basically. Okay, so everything I just said in this video so far was basically the most important part. Like that stuff was the real like foundation of knowledge for the projects we're gonna be doing. And yeah, that's like the foundational knowledge required to start doing some like basic bioinformatics. Now for the rest of this video, I'm going to go a little bit deeper on the things that we just talked about. I'm going to go a little bit deeper on each part and tell you guys some more information about them. And so this stuff I'm about to tell you guys, this is it is important. It is important, especially if you want to like switch into bioinformatics like for your career. It is definitely good to know, but memorizing this stuff won't be important for the purposes of this course. So for the purposes of just this course and the basic analyses we're going to do, this stuff I'm about to tell you guys will be like some extra information. But yeah, the stuff I said so far was the really like important foundation, at least for our first project. But okay, yes, let's go a little bit deeper on each part of this like central dogma process that we just talked about. So like we said, DNA is the molecule that stores the information to produce messenger RNA, which ultimately produces proteins. And so DNA is a molecule that's made up of 4 bases, adenine, thymine, cytosine, and guanine. And in bioinformatics especially, we usually just call these like A, T, C, and G. And the molecule is double-stranded, so there's 2 strands like shown in this picture here. And so there's a base on each one of the strands. So for example, this one here, the one strand has a adenine base up here, this A right here, and then the other strand has a thymine base. And the bases on the 2 strands are complementary in the sense that A and T go together and C and G go together. So you guys can check that in this picture if you want. Like this one is G and C, and then T and A. And yeah, they go together in this complementary sense, so that if you see an A on one strand, for example, the other strand will almost always have a T. I say almost there because there could be some mutation or something, like if there's a accidental mismatch, but those are very rare. And yeah, normally they are complementary in the sense that if you know the bases of one strand, you can pretty safely assume what the bases of the other strand will be. And because they're complementary in this sense, where there's one on each of the two strands, we call them base pairs. So the base pair refers to one of these pairs of bases. Just like what it sounds like, basically. And yeah, but because one strand implies the other strand in this complementary sense, usually if we're talking about a sequence of DNA, a sequence of these bases, the sequence will just be telling you the bases of one of the strands, because the other strand is like implied by that. But yeah, so these sequences of base pairs that we abstract into being these A, T, C, and G letters, these sequences are what encode the information that's used to make the messenger RNA, which eventually is used to make the protein. So like we said before, the messenger RNA is produced from the DNA in a process called transcription. That's why, if you guys remember, the strands of messenger RNA are also sometimes called transcripts. And the way this happens is that, well, for one thing, to tell you guys, the messenger RNA is also made up of nucleotides, also made up of these bases that we just call by their letters. 3 of the bases of messenger RNA are the same as DNA. That's A, G, and C. But rather than having a T, rather than having a thymine like in DNA, messenger RNA has a U, which is short for uracil. And another thing is that the messenger RNA is single-stranded rather than double-stranded like the DNA. But basically the way this transcription process happens is that this enzyme called RNA polymerase binds on to the DNA and kind of goes down it like a zipper almost, kind of unzipping and then like reading one of the strands of DNA. And then it basically puts together this string of messenger RNA bases in a way that they're complementary to the strand of DNA that's being read. So just like the 2 strands of DNA are complementary to each other, the mRNA strand is put together also in this complementary sense. So basically, if it's on the strand of DNA that it's reading, if it sees like a G, it will grab a C nucleotide and put it in that position for the mRNA. If it sees an A, it will grab a U, a uracil. Because remember, we're using that for the mRNA instead of the T's, the thymines. If it sees a T in the strand of DNA it's reading, it will grab an A for the corresponding position in the messenger RNA. So it's basically stringing together these bases to make this strand of messenger RNA in a way that's complementary to the sequence of bases in the strand of DNA that it's reading. So hopefully you guys can see like the totally cool and like amazing thing about this is that it's copying the sequence so that it's also copying the information stored in the sequence. Because we're getting a complementary sequence, we're also retaining whatever information is stored in that sequence of bases, basically. And yeah, here's just another slide on messenger RNA. Again, it's a, oh sorry, I actually forgot to tell you guys like what this type of molecule is called. It's called a nucleic acid. And yeah, both mRNA and DNA are nucleic acids. And yeah, nucleic acid basically just means it's made up of nucleotides. It's like a string of nucleotides. Remember, each nucleotide has these bases that we represent with these letters. The DNA has base pairs, meaning it has like 2 bases, and the messenger RNA is single-stranded, so it only has one base in each position. And yeah, again, there's 4 bases. 3 of them are the same as DNA. That's adenine, cytosine, and guanine. But then it has uracil, which we just call U instead of thymine. And again, yeah, like the mRNA is single-stranded usually. There's some rare exceptions to that we don't need to get into, but there's some like rare exceptions when it could be double-stranded. But for the purposes of this course and just like usually in general, yeah, the mRNA is single-stranded. And it's one of several types of RNA. So yeah, messenger RNA or mRNA is one of several different types of RNA. There are different types of RNA that do different things in the cell. But yeah, this is we don't need to get into that. If you guys are interested in the other types, you guys can research them yourself. But yeah, for this course and for the first project, we'll be talking about messenger RNA or mRNA. And then, so like we said before, the process where a protein is strung together out of amino acids using the information in the messenger RNA is called translation. Yeah, translation is the process of stringing together amino acids basically based on information in the mRNA, where ultimately these amino acids will then be kind of like processed and folded together to make like a functional protein. So the way this process of translation works is that if you guys remember, like we just said, the mRNA is basically like a complementary copy of the sequence of nucleotides in the DNA. So it's basically retaining that sequence information from the DNA, and then the mRNA sequence can be thought of in terms of triplets of these letters, triplets of these nucleotides, where a triplet of letters, a triplet of nucleotides is called a codon. Codon is 3 of these letters in the messenger RNA. And so basically in the process of translation, the messenger RNA strand attaches to a ribosome, and then the information is basically read in terms of these codons, these triplets, where each triplet specifies a certain amino acid to be grabbed and then like strung together in this string of amino acids that's being put together. And if you guys see this diagram over here on the left, this is basically a diagram showing how different combinations of triplets of nucleotides in the mRNA can specify different amino acids. So this outer ring here is telling us a bunch of different amino acids, and then these letters are telling us like how different triplets of letters in a codon can specify different amino acids, or whether to like start or stop the translation process. So something we'll talk about when we do some of the biology for the second project will be different ways that like a 1-letter difference in the DNA between two people could be either like more or less consequential in terms of like the biological impact. So you guys might see, just as like a hint towards that, you guys might see in this diagram that like you could sometimes have like one difference in a letter giving you the same amino acid. So if you have U, and then someone else has like UC, both of those code for the same amino acid. Or you could have a 1-letter difference giving you a different amino acid in that position where that codon is. So you could actually get like between two people, if they have like a 1-letter difference in DNA, that would mean a 1-letter difference in the codon of their mRNA, which would mean possibly a different amino acid being grabbed and put in that place. And then possibly, like ultimately, that could lead to a different biological function of that protein. Maybe it's like more or less functional, or somehow biologically or like biochemically different because of that one different amino acid in that one position. So yeah, we'll talk a bit more about that for our second project, but I'll kind of like hint towards that now if you guys want to ponder over it a bit. And yeah, so just have a slide on proteins here. So like we just said, they're made of these chains of amino acids that are strung together based on the information in the messenger RNA, which if you guys remember was really the information from the sequence of DNA, and the mRNA is like an intermediate step of carrying that information. And so yeah, they're chains of amino acids. After they're strung together in the translation process, they actually undergo some more processing to get like folded together to produce ultimately the 3D structure that is the like final protein. Like we mentioned before, it is possible that different types of proteins can be produced from the same gene. One of the ways that can happen is based on like different ways the mRNA can be like cut up and like spliced together before the translation process. You can get different chains of amino acids based on that phenomenon that's called like alternate splicing. But yeah, that's a little bit like beyond the scope of this course. And yeah, so proteins basically do like the actual work of the cell, catalyzing metabolic reactions, structural support for the cell, signaling, regulating various cellular processes. Again, there's some proteins called transcription factors that actually regulate the expression of other genes or even of their own gene. So yeah, there's a lot they can do, and yeah, they're basically what do like the actual work of the cell. And so yeah, those past couple slides were basically just like going deeper on each one of the each one of the parts of this whole diagram. We talked about DNA, the process of transcription, mRNA translation, and then proteins. And again, just to like repeat, this whole process is called gene expression. So when we talk about gene expression, we're talking about the process where information encoded in the DNA is used to make an actual product, meaning the mRNA or the proteins, and then basically having this information stored in the DNA actually affect the biological function of the cell, basically. And again, just to repeat, like again, different gene expression patterns could be signs of interesting biological differences between cells. So we could ask, for example, what genes are differently expressed between cells that have a disease like cancer or healthy cells. Like what are the differences in gene expression between those different cell states, like disease versus healthy. And also, I tried to do my best with this diagram. I tried to like make this like an informative diagram and have these like slides be useful and informative. But there's only so much I can do in terms of this like my frankly like kind of bad graphic design. So in addition to like looking at these slides, I'm also going to put a link in the description to a really great animated video showing this process of gene expression. So yeah, there's a really good animated video that I recommend you guys to check out to see a really cool like animation of this whole process of gene expression from the DNA to the mRNA to the proteins. So I'll put that link in the description if you guys want to check that out. And I do recommend watching it just to like have it like solidify in your mind this process we're talking about. Okay, there's one more thing I'm going to tell you guys before we like wrap up this video. One more thing that's actually kind of cool and interesting, even though this is beyond the scope of this course. This is epigenetics. Yeah, this is going to be beyond the scope of this course. We're not going to be doing any epigenetic analysis, but I am going to like briefly tell you guys about it just because it is pretty cool and interesting. So we were talking before about this process of gene expression, where the information stored in the DNA can be used to make strands of messenger RNA and then ultimately used to make proteins that do the actual work of the cell and do a lot of like the actual like biological functions of the cell. And so we said before that we talked a little bit about how that process is like regulated. So we said that we can think of it partly as being just a random stochastic process of strands of messenger RNA being produced from the DNA and then kind of floating around in the cell a bit, and then also breaking down and disappearing over time. And then also proteins being produced in a similar like random stochastic process from the mRNA, and then the proteins floating around in the cell a bit, and then also breaking down and disappearing over time. And we mentioned that, like, even though there is randomness to this process, it can be also regulated by the proteins themselves. These proteins called transcription factors can actually like bind to the DNA and then make it easier or harder to have transcription of these other genes. So it can actually like positively or negatively affect the expression of other genes. So that's one way that gene expression can be regulated. Another way that gene expression can be regulated is actually based on the 3D structure of the DNA itself. So we said before the DNA is this like double-stranded molecule. And in complex organisms like humans, for example, this DNA molecule is actually organized in a like complex 3D structure. It's called, in humans, it's called chromosomes. We have like 23 pairs of chromosomes, and they're the like 3D structure of the DNA. And epigenetics basically refers to ways in which gene expression can be regulated based on the 3D structure of the DNA. So one of the ways that gene expression can be regulated based on this 3D structure is just to give like one example, it can be based on how tightly or loosely the DNA is wrapped around these proteins called histones. So we can think about just this like double-stranded like sequence of DNA being wrapped up in this 3D structure, like wrapped up either like tightly or loosely around these histone proteins. And then we can think about then DNA that's like loosely wrapped or that's in between histones is accessible for RNA polymerase to bind and then transcribe those stretches of DNA. But DNA that's like tightly wrapped up around the histones, like tightly wrapped up in these coils, is inaccessible, so that any genes that are on this stretch of DNA are basically being like silenced, so that they can't be transcribed into mRNA, and they can't be expressed in a resulting protein. So yeah, sometimes when people talk about like epigenetics, they like to give a lot of like metaphors. Like they might say that like the DNA is like the sheet music, but the epigenetics is like the way that the sheet music is like being played by the musician. I actually find those metaphors to be sometimes like they just make things even more confusing. So I prefer to like avoid the metaphors and just talk about like what is actually happening at the molecular level, and talk about how it is about how different ways that the 3D structure of the DNA can be organized in a way that makes like some genes accessible to be transcribed into messenger RNA, and some genes inaccessible to be transcribed. So the genes that are inaccessible are basically being silenced so that they can't be expressed and can't actually result in a protein. But the genes that are accessible are able to be expressed and result in a protein. And so one thing that's really cool about epigenetics is its role in cell differentiation. So one thing that's really cool to think about is how in the human body, almost all of our cells have the same sequence of DNA. With the exceptions of like gametes, for example, and the exceptions of some types of blood cells that don't have DNA, or of cells that just happen to have like random mutations. But besides those exceptions, almost every cell in the human body has like the same sequence of DNA. So we can then ask, like, if they all have the same sequence of DNA, how is it possible that we have these like different types of cells that are doing very different things throughout the body? Like we have, for example, liver cells doing a particular type of function, hair cells doing something else, brain cells doing something completely different, and skin cells doing something completely different. All of these different types of cells having different like biological roles and doing very different things. and having like really different like biological qualities, even though they all have basically the same sequence of DNA, other than just some like random mutations that they happen to have. So yeah, like how is that possible? And the answer is that it has a lot to do with epigenetics. So epigenetics allows cells that have the same sequence of DNA to have very different gene expression profiles based on epigenetic regulation of this sequence of DNA. So basically, even if like our liver and hair and brain and skin cells all have the same sequence of letters in the DNA, the different epigenetic regulation profiles of these types of cells can result in different genes being expressed at different levels, and so ultimately different proteins being produced in these cells, based on, again, the way in which the 3D structure of the DNA allows for some genes to be expressed and some genes to not be expressed. And those differences in epigenetic profiles can result in like different gene expression patterns, which ultimately result in different biological differences, even in cells that have the same sequence of letters in the DNA. And the way that this happens in development, the way that we get all these types of cells from originally like embryonic stem cells is through a process called cell differentiation. You might also hear it called like cell fate determination, where the stem cells start off in the embryo as being like just like general cells that don't really have a defined role in the body yet, and then it's still a bit unknown how the actual mechanism of this happens. But ultimately, we end up with these different types of cells that have different epigenetic regulatory profiles that allow for different patterns of gene expression, that allow for ultimately biological differences and different roles throughout the body in these like very different types of cells. So yeah, I just think that's pretty cool. So I just want to like end this video on that note, even though again, we won't really be dealing with epigenetics in this course. It is pretty cool. And yes, that's kind of like the final note on this video on gene expression. Yeah, so I guess we'll end there. In the next video, we're going to get a bit into more of the biological setup for this first project we're going to do. We're going to talk a bit about what the data set will look like for this first project. So that'll be the next video. And yeah, thanks for watching, and see you guys next time.